Welcome back to our series of HRMAn tutorial videos. This video will show you how to use another KNIME workflow to pre-process images that haven't been recorded on a high throughput screening microscope, but on any microscope that is available to you. Let's get started. In this case, I have prepared a couple of images that I have recorded on a Nikon fluorescence microscope. I had infected cells with toxoplasma, mounted them on cover slips, and then imaged two different samples, sample 1 and sample 2. And for each sample, I just acquired two fields of view, area 1 and area 2. The samples had been stained before with DAPI for the nuclei, the toxoplasma that I used for infection express GFP, and then I also stained for ubiquitin and a secondary antibody that is labeled with Cy5, so this would be in the far red channel. Now this is how it looks like, one example image. So we got a couple of cells, some of them are infected, some are uninfected. You can see the toxoplasma in green, and some of the cells will respond to this infection by ubiquitinating the vacuoles. And here, for instance, in this bottom left corner, you can see one of the ubiquitin rings around the toxo vacuole. Now, if you save your data on your microscope, you should save it as a TIFF file and the individual channels. So here's one with the nuclei, one for ubiquitin or the cells, and one with the pathogens. Problem now is, if you do that, you probably name your file something like this. So you indicate what you used for infection, how you treated your samples, how you stained them. The problem with HRMAn is that it was developed to work with images that come from a high content imaging platform. And if you get images from there, the software will automatically name it something like this. So a unique plate identifier, and then where the image was taken. So here, this image would be from well B2, field 37, channel 0, 1, and 2. And this is critical for HRMAn to work, because it will rely on these names to be able to cluster your data and to analyze it. So what we can do, we can go back to our images and name them like this. Now what you could do, of course, is sit down and change the name of every single image you have acquired. But of course that would be quite laborious. So what we did is that we created another KNIME pipeline that you can also download from our homepage, HRMAn.org, put it into your local KNIME workspace, and use it to rename your images. What you need to do to use it is you provide it with a plate map, you execute the node again, and then you can double check. So this should look familiar to you if you watch the other videos. You got three columns with the well, the sample group, and if they were infected or not. For this little software to work, you only need the well names. Then you need to tell HRMAn where to find your images. So you open this node again, you navigate to your directory that contains your images, and then you simply select the folder, choose it, apply, and OK. Then you can execute the node, and our work pipeline will know where to find your images. So now the pipeline knows where the images are, it knows how to rename them, or which well names to use, so you simply execute it. And what will happen is that this box will open and it will ask you how many samples you used. So in this case I used 2 samples. Let's put a 2 here. I imaged 2 fields per sample. So again, put 2 and click OK. And I acquired images with 3 different channels. The pipeline will double check with you. So in this case I got 3 channels, 2 samples, and for each sample 2 fields of view. put a yes, 1 for yes, and click OK. And then the workflow is going to execute, and HRMAn will write your images into the directory where it found the originals. So as you can see here now, you got your old images, and then you got the renamed files in your directory. So these are exactly the same images, they are the same size, uncompressed TIFF files. You didn't lose any information in executing this pipeline and renaming them. Just to prove, so here the first field of view in sample 1 we got toxo, and now this would correspond to well A1, the same image, just renamed. And the same thing then goes for channel 1 and channel 2. And yeah, as a last step, you can delete your old files, and now you can use these images to run them on HRMAn itself. So in this case, if you have used 4 colors to acquire images on your microscope, we're going to use the exact same workflow. I've prepared another example here where I just got 1 sample and I imaged 2 fields of view, this time with 1, 2, 3, 4 colors. Yeah, so what we're going to do is that we again just need to open the workflow and tell it where to find your images. So just again open your directory, here the 4-color imaging, apply and OK it, and then you just simply execute the pipeline. It's going to again ask you, in this case I only have 1 sample of which I took images. I had 2 fields of view, and this time we're not going to add 3 but 4 channels. And then the pipeline is going to double check with you again. So I get 1 sample, 2 fields, 4 channels. That is correct. So I'm going to put a 1 as yes and OK it. And then the pipeline will execute and again write your images into the same directory as where you had found your originals. And yeah, just to prove again, so here first image we got toxoplasma, and this will be now well 1 field 0. So that is it. Thank you again for watching, and we hope that this little tutorial will enable you to pre-process your images so that you can use HRMAn for your own experiments. If you have any problems or any feedback concerning HRMAn or this pipeline, feel free to contact us through our homepage, HRMAn.org.